So we're finishing up part 2 of the From Genes to Proteins PowerPoint. We're starting with from the nucleus to the cytoplasm. So you start with DNA that is then transcribed into RNA. The RNA then does what to make a protein? It must be translated. That translation of proteins will then code for a trait. So in talking about translation, you have to go from the nucleic acid language to the amino acid language. The way we get there is we start off with our DNA nucleic acids, which are then transcribed to our RNA nucleic acid sequence. Those nucleic acids are then read by the ribosome, which will then code for certain amino acids and produce amino acid chains, which are called polypeptides, which will then fold and bend to create our proteins. So how are proteins made? We start off with DNA that's in the nucleus. The information in the DNA in the nucleus is the instructions. We have our mRNA, which has the instructions for building the proteins from the DNA. As we know, the mRNA that comes from the nucleus to the cytoplasm has been spliced and gotten rid of the introns, so now it's all just the exons. So there's no junk. There's no junk in the mRNA sequence. Then we have our proteins, which make a chain of amino acids, and that chain of amino acids will make up a protein. But what reads our RNA? So we need an mRNA reader, which is the ribosome. So what is a ribosome? A ribosome is a structure made from rRNA and protein. It reads the mRNA and translates the mRNA into amino acids. So at the top you have your large ribosomal subunit paired with the small ribosomal subunit on the bottom. So from gene to protein, you start with your DNA to your mRNA through transcription. When the mRNA leaves through the nuclear pore, it is then translated by the ribosome into a polypeptide chain or amino acid chain, which then will form a protein. Proteins are synthesized by ribosomes using instructions on the mRNA. Those proteins then result in a trait. So from RNA to protein, mRNA leaves the nucleus. The mRNA goes to the ribosome in the cytoplasm, and the ribosomes translate the mRNA into proteins. The proteins are made of amino acid chains. But how? So how do you have mRNA code for proteins? Well, you start with your DNA sequence, which comes from 4 different nucleotides, which is your A, adenine, T, thymine, C, cytosine, and G, guanine. It will then transcribe into your mRNA, which also has 4 nucleotides, which include A, adenine, U, uracil, C, cytosine, and G, guanine. But how does that mRNA translate into the protein letter sequence? So how do we get to that? There are 20 different amino acids that are coded in proteins, but how do you just code from 4 different letters 20 different amino acids? Amino acids code for proteins in triplets, meaning 3 amino acids will code for, or 3 nucleotides, will code for a specific amino acid, which will then make up the protein. So you have thymine, arginine, valine, that's asparagine, alanine, I believe, Yeah, alanine, cysteine, and alanine. So when the ribosome comes along, it'll read the first three letters and pull methionine. Look at the second three letters and pull arginine. Look at the next three, valine. Next three, asparagine. Next three, alanine. Next three, cysteine. And the next three, alanine. So these three letters, this triplet, is referred to as codons, which means they code for a certain amino acid. So codons are groups of 3 RNA nucleotides. So cracking the code, Crick determined that there was a 3-letter triplet or codon system. So why did the red bat eat the fat rat? So he determined that for every 3 letters, there was a 3-sequence codon. Nuremberg and Khorana determined the mRNA amino acid match. They added fabricated mRNA, so made up mRNA to a tube of ribosomes, tRNA and amino acids. When they created the artificial UUU of mRNA, they found that UUU coded for phenylalanine. So these are the gentlemen, Marshall Nuremberg and Har Khorana. So here's the code. This codes for all, these amino acids code for all of life. They are abbreviated by three letters of their amino acid. The code is very redundant. There are several codes for each acid. The third base is wobbly, so at times the third base pair may be switched or replaced, and that's okay because the code being redundant allows for that point mutation to occur, but for the same amino acid to be coded for. So in the case of GU, if it's GUU or GUA, you're still going to code for valine regardless. And as I said, the start codon is AUG, which is methionine, and then you have 3 stop codons, which is UAA, UAG, and UGA. So those 3 codons will not code for an amino acid, but will signal for the mRNA to that it's the end of the sequence. So what do we know? We know there's mRNA, which has the instructions for building the proteins from the DNA. We know that proteins are chains of amino acids or polypeptide chains. And we know that mRNA is read by the ribosome. But what brings the right amino acid to attach the protein chain? So we need an amino acid transporter, which is our tRNA. T meaning transporter. Transfer RNA structure has a clover leaf structure. The anticodon is on the clover leaf end. So the anticodon is the opposite of the codon, which is part of the mRNA. On the opposite end of the anticodon is where the amino acid attaches. So this is the 5' end, and this is the 3' end. So loading the tRNA, there's amino-acyl tRNA synthetase, which is an enzyme which binds amino acids to tRNA. The bond requires energy, so you go from an ATP to an AMP. So it's losing two phosphates to gain enough energy in order for the amino acid to attach. The bond is unstable, so it can release the amino acid at the ribosome easily. So how are the codons matched to amino acids? If you're starting with your DNA, you have your complement nRNA, which is has your codons. The tRNA carries the amino acids. So the AUG codes for methionine. The CGU codes for arginine. GAU codes for valine. And these complementary tRNA strands are called the anticodon. So the codon codes for your amino acids. Your anticodon is the complementary strand to your mRNA. So ribosomes, they facilitate coupling of tRNA anticode to the mRNA code. So is it an organelle or an enzyme? If you want to define the ribosome as an organelle, you would be just defining ribosome or organelles not as membrane-bound organelles, but just as any structure that is within the cell. So depending on how you classify it, you may classify the ribosome as a organelle if you're just defining structures within the cell. But if you're defining membrane-bound organelles, a ribosome does not classify. So the structure of ribosomal RNA or rRNA proteins has two subunits. You have the large and the small. The top is the large part and the bottom lighter purple is the small. So at the ribosome you have the A site, the amino-acyl tRNA site. This holds the tRNA carrying the next amino acid to be added. Then you have the P site, which is the peptidyl tRNA site. This holds tRNA carrying the growing polypeptide chain. And then you have the E site, which is the exit site, which is the empty tRNA, which leaves the ribosome and exits from the exit site. So you start the tRNA will enter at the A, proceed to the P, and then exit in the E. So building a polypeptide, you start with initiation, which brings together the mRNA, the ribosome subunits, and the initiator tRNA. There's elongation, so you're adding amino acid bases onto the codons. And then termination is when you get to the stop codon and it ends the polypeptide chain. So protein targeting. Signal peptides give an address or a label to the polypeptide chain. So there are different destinations for the polypeptide chain. It could be secretion to the nucleus, mitochondria, chloroplast, membrane, cytoplasm, etc. So the pathway below shows a polypeptide chain going into the ER. So you have the ribosome transcribing the mRNA. The signal peptide gets an SRP, which is a signal recognition particle. That signal recognition particle protein then attaches to a translocation complex. Upon attaching to the translocation complex, the signal peptide attaches to the complex. The SRP is then removed, and the peptide chain begins to enter into the ER. Once transcription is complete and the the polypeptide chain has officially been removed and entered into the membrane, it will then fold and produce into the proper protein. So can you tell this story from going to the DNA all the way to your um, amino acid polypeptide chain? So you start with the DNA. The RNA polymerase transcribes the mRNA, the pre-mRNA. The exons will be the information that is kept, where the introns are removed. There is a 5-GTP cap on the mRNA with a poly A tail. The reason for the cap in the tail is to ensure that there's no degradation of the genetic material in the RNA sequence that is essential for the protein synthesis. This is the mature mRNA. There's a large ribosomal subunit along with a small ribosomal subunit, which will be the site for the protein synthesis. The ribosome will then read the mRNA strand. It'll start in A, go to the P, and exit through the E. The tRNA, starting from the 5' to the 3' end, is the way the mRNA will be read. The tRNA is going to carry the amino acids into the A slot. The tRNA will also be freestanding, collecting the amino acids needed for the polypeptide chain, and the amino acids are freestanding within the cytoplasm. There is amino acyl tRNA synthetase, which helps add the amino acids to the tRNA, which it'll then help form the polypeptide chain. So from gene to protein, from in the nucleus to the cytoplasm, again we start with the DNA in the nucleus. It's transcribed to mRNA. The mRNA is then translated in the ribosome, and the information is brought by our tRNA, the amino acids are. The amino acids form a polypeptide chain, which is then created into a protein that codes for a trait. Protein synthesis in prokaryotes is a little different. You start off with transcription, so your DNA to your RNA, but the difference is there's no nucleus, so we don't have to add the poly A tail or the GTP head. I'll talk more about this when I get to prokaryotic gene expression. So prokaryotic genes versus eukaryotic genes. DNA in prokaryotes is in the cytoplasm, where in DNA it's in the nucleus. The DNA of prokaryotes is circular, whereas eukaryotes it's linear. In prokaryotes, the DNA is naked, where in eukaryotes the DNA is wound on histones. Prokaryotes are no introns, whereas in eukaryotes you have introns versus exons. Introns and exons allow for genetic variability, so the same transcribed mRNA may code for different proteins and different traits. Translation in prokaryotes. Transcription and translation are simultaneous in bacteria. Since the DNA is already in the cytoplasm, there's no need for the mRNA edit, and there's no need for mRNA editing. The ribosomes will read the mRNA as it's being transcribed. Translation prokaryotes versus eukaryotes. The difference between prokaryotes and eukaryotes is time and physical separation between the processes. It takes eukaryotes about an hour to get from your DNA to your protein, where in prokaryotes there's no RNA processing. Once the RNA is transcribed, it's ready to go and do its job. So any questions, please feel free to post them or ask in class. Also, one question for you to think about and to bring your answer is, what color would a Smurf turn if he held his breath? This is the end of our From Genes to Proteins PowerPoint. Thank you and have a good night.